Bacteria is able to become resistant to the effects of antibiotics by the use of mutations. Here is an outside view of the mutation of cells. Here's a quick look at the key. And here it is. So first up, we'll just start with general mutations. A mutation is a change in DNA that can sometimes cause a change in the gene product of a bacteria cell, canceling out the effects of the antibiotic. It causes efflux or destruction or inactivation of an antibiotic. If the area encoding these genes are mutated, antibiotics are unable to bind to their target, rendering them useless. Mutations can occur due to errors in RNA transcription by polymerase inserting incorrect nucleotides during chain reaction. This happens due to a lack of proofreading ability. This mistake cannot be fixed. Sometimes this can be fatal. It can also cause genetic mutations, allowing resistance to antibiotics. Also, through the process of cell replication, some bacteria develop mutations that allow them to become resistant to antibiotics. The mutated bacteria's genes are then replicated, multiplied, and shared amongst other cells, allowing for overall antibiotic resistance. The first mutation is called destruction or inactivation. Many bacteria possess genes that produce enzymes that chemically degrade or deactivate the antibiotic. It is a type of mutation. These enzymes are created by proteins using the protein translation system. Protein translation transcribes DNA into RNA, mRNA. During this process, mRNA must be translated into a protein. This can be done by a transfer RNA, tRNA, and the mRNA working alongside ribosomes to produce proteins. These proteins then act as enzymes or catalysts to target the antibiotics. So before the antibiotic is able to reach the target cell, these enzymes, which is these enzymes are able to degrade or modify it past the membrane protein. This damages the antibiotic, causing its inactivation or destruction. So as you can see, the antibiotics are unable to move past the enzyme. That is because it is targeting them and destroying it before it can go any further, causing any damage. The second mutation is called efflux. An efflux pump is essentially a big channel that actively exports antibiotics and other potentially harmful components out of the cell before they are able to do any damage. The antibiotic can enter through a small channel called a porin, but it is then pumped back out by the large efflux pump. By pumping the antibiotic out, the bacteria cell prevents it from reaching its target and killing the bacteria. So say that this is an antibiotic. It'll enter through a small porin, but then it'll just be pumped out again by the efflux. That way, there is no harm caused to the cell. Now that we have covered the general mutations, let's talk about how these mutations spread from cell to cell. Here's a general view of the first way that mutations can spread between cell to cell. It's called conjugation, and it's when two cells form a bridge to pass DNA alongside each other. This is an up-close view of how conjugation works. Conjugation begins with a circular kind of DNA present in the bacteria cell, called plasmid, which individually replicates genes and chromosomes for the cell. Here's a non-mutated plasmid, and here's a mutated plasmid. The plasmid is the red part, and the mutated gene is the blue part. Since mutations are present in the DNA of a cell, the mutation would be in the plasmid. This allows for the spread of the mutated gene within the cell since it duplicates all throughout, but in order for other cells to receive the mutation, they have to interact. So when two cells become close, or they get close to each other, the pilus of the cells, which are the red and the pink part, the little strings, form a bridge between the two. And bacteria cells, when they interact, they can share DNA. So since it's been duplicated, this cell sends the DNA across the bridge, and now the other cell has the mutated gene, and it can replicate it all throughout. This transfer of DNA allows for both cells to contain the mutation and to spread it even more. So this is one way that a mutation spreads between many bacterial cells. Here's a general view of the second way that mutations can spread between bacteria cells. It's called transformation, and it's when naked DNA enters another cell. So there's the naked DNA, and then there's another cell. So here is a living mutated cell. It contains the pilus with the mutated gene. It can properly adapt and resist antibiotics. But what happens when this cell dies or breaks apart? The DNA can still live on, so the DNA is released into the environment and free floats, bringing up the term naked DNA. It's called this because it's surrounded by nothing, it's just floating around. It's essentially naked. So other cells can come across this in the environment, and they can infuse the DNA into their own genetics, and now they contain the mutated gene. So since they have the mutated gene, it can replicate all throughout the cell and become mutated. So this allows for more and more cells to become mutated by picking up mutated genes in the environment. So this allows for the further spread of mutations to resist antibiotics. Here's a general view of the final way that mutations can spread between bacteria cells. It's called transduction. Here's a cell, and here's a bacteriophage, and it is going to enter and infect it with mutated genes. Transduction revolves around a virus called a bacteriophage, more commonly referred to as a phage, entering and taking over a bacteria cell, intercepting the genetic processing of the cell in order to produce more phages. So here it is, and it's taking over this cell right here. This causes the bacterial DNA to become incorporated into the phage's DNA. The phage moves from cell to cell, taking genetic properties from each one. But if one of these cells were to contain a mutated gene aiding in bacterial resistance, it would spread just as the phage does. So here's phage spreading. It starts with this cell, it takes the genetic information, and then it takes the genetic information and it infects it to this one. So it gives this one the mutated gene and it enters it through the plasmid. And then this one spreads, and it takes the same genetic information into this one, just spreading the mutation between cell and cell. The phage essentially just brings portions of other DNA into more and more bacteria cells. Even if a phage kills a bacteria cell, the mutation living on in that cell will now live on in another one.